Hello, my name is Jules Savat. I'm a genetic counselor at Geisinger, a member of the ClinGen Education Workgroup, and coordinator for the ClinGen Patient Registry, Genome Connect. This presentation will provide a brief introduction to patient data sharing and will provide information on how patient-shared genotype and phenotype data can inform variant classification. Data sharing is vital to the process of understanding the relationship between genes, genetic variants, health, and disease. ClinGen encourages data sharing from a number of stakeholders, but patients can directly share their information by participating in a patient registry that shares data, such as Genome Connect. Individuals that have had genetic testing enroll online and provide their health history using a health survey. After completing this health survey, participants upload a copy of their genetic testing report so that our genetic counseling team can collect information about any genetic variants that were found. A participant's genetic and health information is then de-identified and shared with NCBI's ClinVar. Phenotype information from participant health surveys is mapped to human phenotype ontology terms, allowing for structured data sharing. In terms of genomic data, the variant, clinical significance reported by the laboratory, report date, reporting institution, and associated conditions are collected directly from the participant's report, and these data are submitted as a phenotyping-only submission. This submission type highlights that Genome Connect does not make an attempt to interpret the variant independently, and makes it clear that the laboratory that completed the participant's testing may also submit to ClinVar. Once this information is available, how can it be used to inform variant classification? From our experience with Genome Connect, we recognize that patients opting to share their information are an important source of genomic data that otherwise may not be publicly available. The more we aggregate genomic data, the better we'll be able to classify variants. It is clear that the more a variant has been identified, the more we are able to understand if that variant causes disease. More than that, if you examine the ACMG sequence variant interpretation guidelines shown here, you can see that even if a variant at the same amino acid residue has been identified previously, variant classification can be impacted. In this way, patient-contributed variants can also contribute to the understanding of other variants. Second, patient-provided data affords additional case-level information that can be used to inform variant classification. Laboratory submissions are often in aggregate and may include a few phenotypic details. Patient-provided data is case-level rather than aggregate and often includes additional phenotypic information and provides the ability to recontact over time for additional data or as health changes. Patient data includes segregation information if available and can inform if any other variants were detected in that individual that could explain their phenotype. This case-level information can be used to inform classification in a number of ways highlighted here. For example, Genome Connect submitted this SCN8A variant. The variant was interpreted as likely pathogenic by Lab 2 in 2015 on the participant report. The variant was also found to be de novo through that testing. Ultimately, Lab 1, another laboratory that had identified this variant previously, reached out to the Genome Connect team to inquire whether maternity and paternity had been confirmed during testing and whether the variant was in fact de novo in our participant. This segregation data allowed Lab 1 to update their classification to likely pathogenic as well. Finally, Genome Connect facilitates connections to inform gene-disease relationships. Genome Connect submits variants and genes of uncertain significance to Matchmaker Exchange, a federated network of rare disease datasets established to facilitate candidate gene matching. It is necessary to understand whether a gene is associated with a specific phenotype before a variant can be classified, and Genome Connect contributes data to these efforts through Matchmaker Exchange submissions. So how can you leverage patient-shared data to inform your classification efforts? First, review ClinVar. Variant information from Genome Connect participants or other registries engaging in data sharing is submitted to ClinVar periodically. We are happy to answer questions regarding our submissions, including whether other variants may have been detected in that individual, whether maternity and paternity were confirmed when the variant was reported as de novo, or to invite participants to participate in functional studies or additional research. In addition to providing information that can inform variant classification, patient data sharing allows patients to remain up to date regarding their genetic test results. If the Genome Connect team learns that a participant's report is out of date and the reporting laboratory has updated their classification, this information is provided back to participants that elect to receive it. To summarize, Genome Connect offers a structured way for patients to provide additional phenotypic information, case-level data, and novel genomic information to inform variant interpretation and genomic medicine. Genome Connect is currently submitting patient-provided data to ClinVar and is working to enable other registries to do the same. You can find a list of registries recognized by ClinGen for their data sharing efforts here. I hope this has been a useful introduction to patient data sharing and its potential impact on variant classification. Please visit www.clinicalgenome.org for additional educational resources and to learn more about ClinGen initiatives. Our funding sources are listed at the bottom of this slide. Please feel free to contact ClinGen by emailing clingen@clinicalgenome.org or contact the Genome Connect team directly at info@genomeconnect.org with questions or feedback. Thank you.